Hey guys, Mr. Kate again. This is Chapter 9 Notes, Comment Part 2. And so we'll begin with slide 15. We ended with slide 14 last time, so we'll take it right up and get going here. On this slide, there's a lot of stuff going on in here, and this is all about the Krebs cycle, where you are inside the mitochondria now. You had the acetyl-CoA piggyback the pyruvic acid. First molecule it turns it into is citric acid. And then it starts manipulating this molecule over and over and over again. It just rips off carbons. As it does that, every time you break a bond again, remember it releases lots of energy. So it's breaking carbon off of there. It's breaking off hydrogens. The hydrogens are picked up by the NAD pluses and by the FADHs during these processes. And the carbon is then bonded. Some of that energy is used to bond oxygen with the carbon. So it binds the carbon immediately so it doesn't try to rebond with the citric acid that it was originally ripped off of. Okay, so here in this little paragraph, it says during the Krebs cycle, molecules are being broken down and reconfigured. Why? And explain what is the end game or process, or what is the process trying to achieve. And basically the process is trying to achieve by the manipulation and the ripping off of these various elements is to, one, capture the hydrogens, and two, that released energy from those bonds is captured to bond CO2 together, and also make and manufacture these other new molecules. And what's left over then is used to manufacture a couple of ATP molecules. Okay, so it's like three different things that it's being used for. All right, and moving on to the next slide here. This one here is, where does the energy come from to produce all of the NADHs and FADH2s? It goes back to that last slide I just talked about. It comes from the manipulation and the breaking of the bonds from one molecule to another in the Krebs cycle. So as that Krebs cycle is changing those molecules, going from citric acid to all of the other different kinds of molecules, every time it manipulates that, takes off a hydrogen, then it will capture that hydrogen with the energy carrier, the NADH, forming an NADH or an FADH2. And as it's peeling off those hydrogens or peeling off those carbons, it's releasing energy, and that energy is made to bond those molecules all together. Okay. This slide here is just asking you how many ATP are actually made during the citric acid cycle or the Krebs cycle. And again, if you forgot, go back up to page 3 and it will show you. So go back up here and look over here in the chart, and it will show you how many ATP are actually generated. Okay, so that gets us to this one. And here again, you would grab, drag, and drop it into place. Okay. Actually, it should be this one here. Sorry. And the next slide here is just a summary slide. So you're going to write a summary here on the right side using the caption box, or down at the bottom using the summary text box. And what is the ETC? Basically, it's just the inner membrane that has several protein channels embedded in it, and it's going to be taking this released energy and moving it through the membrane. It's going to take the hydrogens that were gathered from the NADHs and the FADH2s and move those across into the inner membrane, across the inner membrane to the inner membrane space. So it's that space between the two membranes. All right. Oxygen must be involved in this process, otherwise the ETC would not work. All right, and we'll see that in a slide coming up. Okay, so on this one here, again, you're going to summarize the slide. You're going to put a summary over here on the right or the summary statement at the bottom. The electrons are passed between the carrier proteins, and eventually you're going to be generating some ATP. Okay, so in other words, this slide here is just another reiteration or going back over what the last slide just stated also. All right, this slide here is the one I was waiting for. Here you're going to actually visually see that the NADHs are releasing their energy. The energy is traveling through the membrane itself and being released over here and used to bond the hydrogen and oxygen together. And also the hydrogens are being driven across into that inner membrane space. Now these proteins here drive them across. ATP synthase is a one-way valve going into the matrix, which is this space down here. Okay, so this is the space inside the mitochondria. This is the space in between the mitochondrial membranes. So the buildup of H plus ions is in between that inner membrane space. And because you have lots and lots of H pluses over here, but very few over here, any opening that there's available, these things are going to flow through and go into this matrix area. And as they do that, they're going to take their high energy, and that high energy is now going to be put to use to form ATP. It's going to bond a phosphate and ADP. It's going to bond them together, and now you've got a loose hydrogen over here. Well, if you don't do something about this hydrogen, the number of hydrogens here is going to equal the number of hydrogens here. If that happens, no ATP is going to be generated because the hydrogens are not going to flow from here to here. They flow from high concentration to low concentration. If the two concentrations equal each other, that flow stops. So does the production of ATP. So that's where oxygen comes in. Oxygen comes in, will take this hydrogen, bond with it. Okay, it uses the energy that's over here, takes that energy, bonds that hydrogen with the oxygen, and once it's in the form of water, it is no longer a threat or a variable that's going to keep hydrogen from flowing through that channel. Okay, it has to be like things. So this one here is basically asking you to answer this particular statement. So it says the yellow line represents the pathway of electrons, and H plus ions are forced through the ATP synthase protein channel to form ATP. And I did reword the last sentence here. Where does the energy come from to form ADP plus P to basically manufacture or make ATP? Where does the energy come from? It comes from all of these ions in high concentration being driven through the ATP synthase itself. Okay, so that's the answer for this. All righty. That one took a little while. All right, number 21. Again, you're going to do a summary on this one. A summary on the right or a summary at the bottom, where CO2 is going to be given off in the Krebs cycle, water is given off in the electron transport chain. Okay, so water is a product of the electron transport chain, where carbon is given, carbon dioxide is given off in the Krebs cycle. Okay, and how many ATP are produced in the ETC? And again, go up to page 30, to page 3, not 30, go to page 3, and you'll notice it's like 34-ish. All right, a couple more slides here. So we've got another summary here. And this set of slides from this point on is what happens when there's no oxygen available or not enough oxygen available. All right. Well, what happens is the system takes over from manufacturing just an enormous amount of ATP to having to compensate and make ATP via glycolysis and glycolysis only. And it keeps that glycolysis running really fast. I mean, it super speeds it up. But in doing so, it takes pyruvic acids, which are going to change into lactic acid. And as it does that change, it's going to produce a lot of ATP. But the lactic acid has a downside to it. It will and is detrimental to your muscles. So if you continue to produce ATP through that method, it's going to be very detrimental to your muscles. In other words, it will hurt your muscles. Okay, so it is anaerobic, meaning no oxygen is used. And when all the oxygen is used up, then it has to switch over to this method. Okay, next slide here. If no oxygen is present after glycolysis, then it's going to go into fermentation, and that's where it's going to start producing this lactic acid. All right, so no ATP is made during fermentation. The ATP is made during the break apart and breakdown of glucose. Okay, that's where the ATP is made. Now, what happens to all these NADHs that are being formed during glycolysis? Well, the H's are being spun off and reforming a new molecule, that's the lactic acid. Okay, so you got to have these H's disappearing and being used somewhere else so that it can go back over to glucose and gather up more hydrogen that are being broken off as it's being broken down to pyruvate molecules or pyruvic acid. Okay. This one here, glycogen, is a string of glucose. It's normally stored in the liver and in the muscle cells. And glycogen is a high energy set of glucoses. In other words, it's one glucose molecule strung right next to another, strung right next to another. And when you start breaking these down, you can generate a tremendous amount of ATP this way. But on the other hand, as you generate a tremendous amount of ATP, lactic acid is building up, building up, building up. So you can only do this for so long before you start paying that major price. Okay, this one here, I just want you to find a picture that of some organism that represents lactic acid fermentation. In other words, they produce lactic acid fermentation, and some organism that produces alcohol fermentation. Okay, you do not need to summarize this. You just need to find pictures and place them here on the right. And almost done here. Here you have lactic acid fermentation building up, building up, building up, and as it builds up, your muscles are going to get really fatigued. They're going to get achy, tired, burning sensation, and this is all because this acid that's near and on your muscles is basically irritating them a lot. Okay, and there's no oxygen there. Now, oxygen, once you get oxygen back in your system, and you're breathing real hard, but you stop doing your exercise, whatever you're doing that puts you in that situation, and which is called oxygen debt. Once you start getting enough oxygen back in your system, you start repaying that debt. The lactic acid will start being broken down by the introduction of this oxygen. Okay, and this one here, you just need to name a bacteria that is used for fermentation. So look one up online and type it in here. And there's a few of them. All right. This slide here, you can see it's got stuff all around the borders, and all of these words you're going to use to plug into the boxes and to the little stars. You're going to label the arrows. You're going to label the brown area